Hello and welcome to BrainCast. Today I'll be speaking to Corne Kros, a Professor of Neuroscience here at Sussex. After completing a degree in medicine from the University of Groningen in the Netherlands, Corne began a PhD at the University of Cambridge, during which he developed pioneering techniques in the field of hearing research. Since then he has led an extensive and prolific career in this area. More specifically, his lab attempts to find ways to protect cells in the inner ear from dying, and in doing so, prevent hearing loss and deafness. Today we discuss this fascinating and important research, but also get some insight in what it's like to be an academic, and hear some advice for those aspiring to do a PhD and begin a career in research. Thanks very much for coming. It's very kind of you to lend us your time.
Thanks for the invite.
We just wanted to have a bit of a chat about your research and that kind of thing. You and I haven't actually met in person before. We only met via Zoom.
Yes, indeed.
I suppose it's how you meet most students for the last few years. Has been quite a lot of Zoom.
Indeed, I'm very glad to be moving away from that and see people in 3D. And,
yes,
exactly.
After years of lecturing in real life, it must be such a strange adjustment to suddenly everything going online and things.
It was. It took a long time, and it takes time to get back to normal as well.
Yeah, I can imagine. We'll just start really with at the start of your career.
Mm-hmm.
So you started with a degree in physiology from Cambridge.
Yeah, that was my PhD basically.
So your BA and then you went and did medicine. Was it at Groningen in the Netherlands? And then came back to Cambridge for your PhD. So it would seem to me, I might be wrong, but neuroscience maybe wasn't initially your first sort of choice.
Mm-hmm.
So how did you sort of become interested in neuroscience and your field of interest specifically?
Hearing, and ototoxicity. So neuroscience. I actually started. I'm Dutch, and I started my academic career studying medicine in the Netherlands, in Groningen, in the north of the Netherlands. Mm-hmm. I really liked physiology, I realized, and it sort of turned out that so my physiology professor was an Englishman. Mm-hmm. I sort of talked to him, can I do something more about physiology? By physiology, I think I meant mainly neurophysiology, because I just found the brain amazing. I remember as a medical student buying my atlas of the medical anatomy atlas, and just thought the brain was so much more complicated than any other organ. All these weird Greek names like amygdala and hippocampus and stuff that I had no idea what they meant, and I just thought, I want to find out about that. That's really interesting. So that's that. I started in medicine, then I sorted out that I could do a year. I did part 2 physiology, a year in Cambridge to do physiology, and then indeed I finished off my medical training and then went back to do went back to Cambridge. Hearing, I love music. Music is one of my it keeps me going really. It's really important for me, and when I was in my teens, I sort of started listening to exploring classical music, and I was listening to on headphones because I didn't want to disturb my brothers and the rest of the family with my music taste. They were into Pink Floyd, I was into classical and stuff. So I was listening to the Military Symphony by Haydn, number 100, and it's got a triangle in it, and I realized when I turn around my headphones, the triangle is still on the right side. So I thought, there's something wrong with my hearing, and I was quite worried about it. So I went to an ENT doctor who tested my hearing, was whispering behind me and did all the hearing tests. And he told me at the end, geen zorgen voor de dag van morgen, so no worries for the day of tomorrow in Dutch. But clearly, there was something not quite right about my hearing. So I thought, that is kind of interesting. What's happening there, basically? Mm-hmm. So that's the start. The interest in hearing is really an interest in music, and by no means being a good enough musician to pursue that, mm-hmm. and being quite a decent student of I think medicine and other aspects. So I thought that's what I go for. So then in Cambridge, with the PhD, basically I chose the supervisor who really did inspiring research in the area of hearing. So yeah.
Right. Yeah, I think neuroscience is one of those things where it really allows you to pursue a passion you have outside of science, because everything interfaces with the brain, really. So something like music, obviously, is entirely fascinating in and of itself. But to be able to study how we perceive it on a biological level, I can definitely understand why that would be.
Yeah, it was really intriguing, and I'm still learning every day, basically.
Yeah,
so,
yeah, of course, yeah. So, what did you choose to do for your PhD? You said that you picked a supervisor who's inspiring in hearing.
Yeah.
So briefly sort of summarise what you looked at.
So very briefly, in my supervisor's lab, they were working on hearing in turtles, because they had a very accessible ear, and you could take it out and keep it alive outside the body. You could look at the ear and also the hair cells. They were very interested in the hair cells that just use sound into electrical signals to the brain. And when I came to chat with my prospective supervisor, he basically told me, do whatever you like. As long as it's within hearing or the vestibular system. I remember going to the pub and sort of discussing that. Mm-hmm. And so we arrived at that because I was a medical doctor, or was training to, I was a medical doctor at that time. I went for the PhD and just qualified. It would be fun, really interesting to try what they were doing on the turtles on a mammalian system. So that's what I did. So I studied hair cells in mammals, in guinea pigs, as it turned out to be. So, and I really did the pioneering work on finding out aspects of how the inner hair cells encode sound. I did the first patch clamp experiments on the inner hair cells. That was really the gist of my PhD.
Yeah, so something quite pioneering then, because I know a lot of PhD theses get printed and then sort of get shelved and never read again. But I suppose you must be quite proud that you were able to do something with your PhD that was sort of groundbreaking.
Yeah, that was great. We got a paper out towards the end of my PhD in the Journal of Physiology, which is still very well quoted. It's got I think pretty much the only picture in the literature of an isolated inner hair cell. It's a really beautiful picture that I saw it at some point while I was doing the experiment. I thought, that's such a beautiful cell. That's much more beautiful than what I normally see. So what shall I do? Shall I try and make another recording, or shall I take a picture? And I just decided to take a picture of it. And the typical experiment that you show in the paper or pictures always, of course, really means the best really that you've ever seen. So
nice, good. Well, that's great. But as I'm sure all PhD students find, it's never easy. So I'm just sort of interested in, was there any particular aspect of your PhD that you found particularly difficult, as you were going through it? Was there anything that stood out as more challenging?
Yeah, it was actually, I would say, the first 2 years of my PhD, quite frankly, just because I was really trying to something pioneering, something completely different. It was actually very difficult to take the cells out and keep them healthy, keep them going well. I had to discover all the tricks, all the ways to do that. So the first 2 years were really quite difficult because nothing worked basically, and then suddenly things started working. I remember, I think it was the 17th of August 1986. It's suddenly I managed to record from the inner hair cells, and I called my supervisor and his colleague, and they started twiddling knobs and stuff on the patch clamp amplifier, and my supervisor basically told me, well, you got your PhD, you just have to do this another 100 times or something like that. So there's the waiting for the breakthrough is just really hard, because you just somehow I kind of lost my confidence in many ways. So that was difficult.
Trying to get it going before you can do much else.
Exactly.
I suppose that must be especially difficult when you're doing something pioneering. Because you can't really turn to anyone for too much help.
No, that's right. You can try and draw analogies and exactly, indeed.
Yeah.
But,
would you, if you could, theoretically, give your younger self, your pre-PhD self, a piece of advice, or in general to any prospective PhD students, what sort of advice would you give, do you think?
Yeah, so in retrospect, I actually think I'm really happy that I went for a supervisor, met a supervisor who told me, basically do what you like. I think that is something, I know often, depending on the lab setup, it's actually quite difficult because you slot into something. I mean, science has changed a bit, it's become bigger scale in a sense. But try and at least steer things into a direction that you find personally interesting, because that actually got me through in the end, also knowing that, yeah, I really care about this and I really would be very proud with whatever comes out of this. The other thing that I sort of discovered halfway through is get yourself something else to do. I started playing recorder and then later flute while I was doing the PhD, just to have some completely different focus and keep a sort of balance and mental sanity going. So
yeah, I definitely think that's important for kind of, any kind of study, is to have some kind of completely separate thing to be getting on with.
Yeah.
And it's interesting that you mentioned the importance of choosing something that interests you, because I think a trap that some people fall into is doing a PhD because they want to just continue in academia and sort of see it as a job sort of prospect rather than a sort of real dedicated.
Yeah, exactly. And I think it doesn't work very well. You really need it. It is hard. It's a really probably the hardest time of your scientific career in a sense to do the PhD. So you got to like it, otherwise, you're not going to be very happy, I think.
Yeah,
and also the people around you probably not. So yeah.
Yeah, this is especially important when science is so collaborative. You really don't want to be difficult to get on with.
No, indeed, correct. Yeah.
So after your PhD, you were at Sussex as a research fellow for a bit, and then to Bristol for some years, and then back to Sussex. So when you first started out as a postdoc doing postdoctoral research, what was something that kind of you weren't expecting, something that caught you off guard a little bit about what it was like to be a postdoc?
Yeah, in a sense, it wasn't that hard, because actually, having done the PhD, which was hard, there were a lot of challenges. I found the postdoc actually enormous fun, because I was came to the lab here in Sussex with Ian Russell and Guy Richardson, who were there at the time. They ran a fantastic hearing research operation, and again, they basically told me do what you like. so I started working on a new preparation, cochlear cultures, that Guy Richardson had been developing, and there was so much to be discovered. It was just nobody had done electrophysiology on those, so I just needed to work out how to record from them and everything, and that actually worked beautifully. I had to learn to work with other people, because the PhD was very much a solo effort. So, but again, I had a colleague, Alphonse Rusch from Germany, who's long died actually of HIV/AIDS. But he was great to work with, and I learned to work with others, and we really sort of spurred each other on to do an enormous amount of work. I think I filled a lab book every 3 months or something like that. Whereas nowadays, I think my lab book dates from 2012 or something. So it's about 3/4 full. So.
A bit less prolific writing.
Yes, indeed, exactly, absolutely, yeah.
So a postdoc you found to be less perhaps scientifically challenging, but more focused on collaborating with others. Also I found discovering work-life balance, keep your partners in life and things like that happy. I was working like crazy all the time, and I sort of had to adjust that also, in a sense. I think everybody has to decide for themselves what is a healthy lifestyle in that sense. I remember I was working in the weekend sort of pretty much all the time, because we were trying to follow the development of the hair cells from day to day as they change during
during development. There's lots of changes going on there.
So you're in there quite a lot, making sure stuff was going.
Yeah, exactly.
Yeah. Okay, well, so that's the start of your kind of postdoc career. You've obviously been established for a long time now. Would you mind giving us a kind of brief overview of some of the most exciting projects that you're working on at the moment in your lab?
Yeah, so currently we are interested in ototoxicity by aminoglycoside antibiotics and cisplatin, and we discovered in the lab some 15 years ago, maybe slightly longer, that aminoglycoside antibiotics enter the hair cells through the mechanotransduction channels. So they're the very channels that are in the hair bundles and that open and close in response to sound. So they're like a Trojan horse that go through those channels. They're not meant to go through there, but the channels have quite a big pore, actually, as people have found very recently. They enter the hair cells, and it's a one-way system. They enter, they can't go out again, really, because the kinetics and the energetics of going through the channel are very favorable from the outside and not from the inside. Mm-hmm. So we found out how that works, and because we know that they enter through the mechanotransduction channels, in collaboration with Guy Richardson, we've been working for years on trying to find compounds that also block the mechanotransduction channels and that you could give at the same time as the aminoglycoside antibiotics to block their entry into the hair cells while the antibiotics are needed, and then as the antibiotic treatment is no longer needed, you can hopefully wash it out and prevent the ototoxicity. So that's the sort of main thrust of our work at the moment, and we found actually a compound that works in the cochlear culture, so in the in vitro preparation, and that also protects the hearing of mice against aminoglycoside when you treat them with aminoglycoside. So you put in one ear, you put the protective drug, and that's with my long-term postdoc in the Richardson and my labs, Richardson and Kros lab, Richard Goodyear, who's been working on that to find that that it protects. So that's great. So we moved the step from in vitro to a real mammal where you can protect. So that's exciting.
Great.
And we're continuing to work on different aspects of that. One thing is that with cisplatin, the evidence is much more murky. It's not very clear. There may be multiple entrance routes into the hair cells. Cisplatin is an anti-cancer drug, for people who don't know that, and it's quite successful in many cases, like for example young men with testicular cancer, which is a disease of young men. The cure rates are very high, over 90%, so they'll have a healthy and happy life in front of them, but quite a few of them will have hearing damage due to the cisplatin treatment for the condition. Mm-hmm. So we're very interested in following that. There's some evidence that it may go through the transduction channel, but there's other evidence that it doesn't. And we currently have a grant funded by the RNID, which is a hearing charity, to find out if a type of ion channel called a VRAC channel, a voltage regulated, a volume regulated anion channel, is perhaps involved in letting cisplatin into the hair cells. So we're exploring that, and it might also be involved in some kind of repair from the aminoglycosides, some defense against the aminoglycosides. So we're studying that as well.
Mm-hmm. I'm just going to check that the audio.
Yes, absolutely. Yeah.
I need to give it a pause, just because it's turned away from me.
Yeah, good. Yeah, exactly.
Okay. I'll try not to move too much from where I was. Yeah. So that's great. That's really interesting. I think it must be quite rewarding to be studying something that has such translatable medical, impact. The fact that you could save a lot of people from losing partially or totally their hearing, it must be quite a rewarding thing to think in the future. Your work could really go a long way.
Could be carried forward effectively in hopefully a direction that helps people. I think that's I've always been mindful. I kind of started out my research in really quite basic science, really the basic aspects, because nobody knew anything about mammalian hair cells and their physiology really. So and I've moved more towards application. I think that it fits with my background, that I managed to do that now. The research climate, of course, has changed in a way as well. That's well funded, really. So yeah.
Well, that's I suppose as well. There's a link to your medical training as well. Long ago, when I should say, when you originally thinking about medicine, and now you sort of ended up doing something that has quite a lot of clinical applications. So it must be quite nice that it has that sort of circularity.
Yeah, I find that yeah, exactly. That's right. It's always frustrating because it's research. Just you never reach the end goal. Really, there's always something. The next step that needs to be taken. So yeah,
there's always more to be done, I suppose.
Yeah, exactly.
Well, related to that, I mean, obviously, your work sounds fascinating. You're making a lot of good progress, but as with all science, there's going to be some element of budgetary constraint applied there.
Indeed.
So, as sort of a fun hypothetical question, if you were to say have 10 times the budget that you have right now, what sort of things would you like to do with that? Maybe what would you do differently, perhaps, or what things would you explore?
Basically, if I get give me 100 times the budget, and then really see through to go from this compound that we discovered that works in the mice onto moving towards clinical trials. It's a very expensive process. It's a very involved process. It involves lots of quite humdrum testing in a way, safety testing before you even go on to the people. Quite rightly so. But I would try and do that and really see that through.
So given that obviously clinical trials are as expensive as you say, and we aminoglycosides, but especially with cisplatin, which is such a commonly prescribed drug to cancer patients, do you ever get frustrated that there isn't more funding for sort of hearing loss research, or perhaps do you ever wish that there was more kind of awareness of the research that you're doing here?
Yeah, it's always an issue, and I think the RNID are doing a great job actually to raise awareness and to get funding from people. But it's usually it's people who have a personal interest, probably because of their personal history or family history around deafness, and we're extremely grateful for people who contribute to the research. It'd be always great to have more research council funding also. That is hard to come by, of course, and the climate isn't easy, the funding climate generally. So more is always better in that respect.
Of course. Well, interestingly, as you'd mentioned before, how when you originally started your PhD, it was much more of a kind of individual sort of sort of do what you're interested in.
Yeah.
Whereas now, sort of big science is a lot more sort of regimented, sort of structured in what people do. I was interested in if you could think of anything that's really fundamentally changed about how you do science, how you do your work since you started your career. I mean, obviously, with an accomplished career like yours, it's been a long time. So I imagine lots has changed. But is there anything you think that's really fundamentally different?
Yeah, I guess I've kept quite old-fashioned in a way. So I still like to have a good idea about what's going on in the experiments and have an interest in the. I'm also very interested in the detail of everything. Right. So I've always run quite a small lab actually on purpose. Colleagues of mine are running much bigger labs, which I think can be very good actually, because it enables you to answer questions in more detail and in a broader range. So I may not be the best person to ask this, but I think actually the expansion into bigger labs and the availability to plug into really a huge range of techniques to answer questions. I think that's more and more the trend in a sense. There's a big change there. I try to sort of do that by collaborating with people worldwide who can offer various areas of expertise that I don't have myself. So that is a big change. I think another aspect is really that is becoming difficult in a sense is managing a career at a university where you do teaching as well as research and balancing that and balancing the time. And at some point that actually becomes very difficult, and that has become more difficult since much more difficult since I started. I had a lot more time to for years I was doing experiments every day myself, and nowadays I'm more a problem solver. So if there's a problem in the lab, I find it really interesting and to try and solve it basically. But I just don't have the continuous time to be engaged at that level at which I started off really.
So you'd say maybe that the sort of the level of kind of admin load and teaching demands is definitely more than when you started, do you think?
It is more than, yes, it is definitely, yes, it is more than when I started, and I think it's something that needs to be kept under very close review really, just as an enterprise and as the way universities work. It's a difficult one.
Mm-hmm. Do you think part of the solution to that would be more lecturers that focus only on teaching to allow researchers to have more time for their research, or do you think there's other solutions?
I think it probably helps in a way, but I've always found, as a consumer, as a student myself, I managed to be a student until I was 31, because medical training is quite long, then a year in Cambridge, then the PhD. Teaching is exciting, where you feel it's informed by people's experience. I got taught by Alan Hodgkin, who was one of the Nobel Prize winners in physiology for working out the action potential conductors in the squid giant axon, and he was working on photoreceptors and vision at the time, and it was exciting. It was absolutely exciting. You could go to him and ask questions, and that was amazing. So, and that may not be the case if you get somebody who's really totally focused on teaching only. Maybe a bit of ideally I would say maybe change the expectations a bit of how much we put into teaching and how much admin we put into there. Effectively try and think what students really find interesting, what they appreciate in teaching, and maybe trim down workloads a bit for people in that way, to make it all manageable. Make use of the technology that is available. So now, I mean, from the whole COVID episode with the Zoom and things, we have learned things, and we have learned to do things perhaps more efficiently than we used to do before.
Mm-hmm. Yeah, I think that's really interesting, because now, like you say, we have the facility where lots of lectures are recorded. You could in theory record, with some lectures where things aren't going to change massively from year to year.
Yeah.
You could just have a recorded kind of bank of those, and then leave time for researchers like yourself to have more kind of engaged discussion.
Something exactly. Just try and be creative in finding out what is really what students really want. And exactly. Yeah.
Well, relatedly as well, I was going to ask next, what is it that you enjoy the most about teaching, and what is it that you find the most difficult?
Okay, yeah. What I enjoy is I love interacting with students. When I give a lecture or whatever, I like to ask questions, and I welcome questions from the students. What I've really enjoyed the most looking back, is projects, the year 3 projects. And basically I have a broad area. I have the area of hearing. I do something in my specialist area, but in my project I also try and work a bit more on my music interest and my interest in deafness. So I always have projects around cochlear implants and music perception, for example, because it's quite difficult. They're very good for speech, but not very good for music. And I always tell students, if you have an idea for yourself in this area, do it. And I've had a student this year who came up with comparing amusia, where people can't hold a tune, but otherwise their hearing is perfectly fine, with hearing and cochlear implants. And I thought that was a fascinating idea. I wouldn't have thought of that. A student came up with that. There was another student some time ago who decided to study tinnitus in zebrafish and to find a way to find out about it. And she subsequently did a PhD with me on aminoglycoside antibiotics. But I had another student who interviewed doctors in Tanzania about aminoglycoside use, and so just to see these people taking initiatives and enabling that's been fantastic to support them in that. So I really like that. What I don't like so much, as a kind of contrast by that, is having to mark people's work. I kind of understand why it's necessary, but I think the real essence of going through an academic study is to find something, to find some sort of purpose in your life, something that really fascinates you. And I think having to when I'm marking people, I may be marking them on stuff that are actually not really that interesting, and they're great at other stuff, and I have to be quite tough really, because it doesn't fit with my criteria. So yeah, that's something that I'm not so happy with. Effectively, yeah.
I think that's a nice. It's nice that you get to see with projects. You get to see the real start of people's academic career. I think, because up until that point, you don't have. You might do laboratory kind of experiments in your time at university, but usually when you get to your project is when you first have that kind of ability to pursue things that interest you.
Yeah, exactly. And I always try to encourage people to really try and think for themselves what interests you and how you're going to go about it. And I'm often amazed and really impressed what people can do.
Yeah, nice. So as a kind of final sort of the one of the fun questions we have to ask everyone in neuroscience, do you have a favourite brain area, and if so, what is it and why?
Right. Well, you may not be surprised, perhaps. It's the bit between the ears. so how the brain processes not just music, but sound and different and speech and it doesn't cease to amaze me. I remember I actually found that at the beginning, I kind of, one of the reasons I went for the ears, that I often say I found the brain a bit too complicated to study physiologically. The ears you can access, it's all beautifully laid out. But for example, in the auditory brainstem, there's all these nuclei, and they process all different aspects of sound. And my PhD supervisor used to say, think of it as a sack of stones, and it's sort of, you can rattle them about, and they'll probably still be stones, but they're all doing different things, and it's difficult to work out which one is doing what. It's more tricky than the visual system, that the steps are clearer. So to try and find out and follow the sort of research of how sound is processed in its details, how you extract information, auditory illusions, things that show that there's other things going on than just what gets in through your ears. That does fascinate me. I've been actually diagnosed now with that one ear that in the beginning, that when I was in my teens, found out that I couldn't hear the triangle in. If I test that ear, I've got hearing loss in that ear, and the other ear is fine still. So in principle, I have a fascinating experiment going on all the time, comparing a good ear with a hearing loss ear. So yeah, and it also makes you think that functionally I've absolutely no problem. So the brain must be able to do a lot of making up for input that is not particularly wonderful.
Yeah, it's impressive that the amount of sort of compensating the brain can do for damage to the organ itself.
Yeah.
It shows how much the brain is responsible for that information beyond just what comes in. So much processing.
Exactly. I think the brains are just. It almost doesn't matter what you put in. It's geared towards making sense of whatever input you put into it effectively. And, yes.
Yeah. Do you think do you ever sort of find it cosmic irony or something quite humorous that someone that's dedicated their career to studying hearing and hearing loss, it has some element of that, or is that just interesting?
I kind of in some ways it started my interest because I was worried about it, and although the ENT surgeon said don't worry about the day of tomorrow, I always kept thinking that's maybe you're not quite right. You're just trying to make me feel good about this, basically. So as a doctor should do sometimes, perhaps. But yes,
it's fun that it's the thing that sort of started off your interest and sort of kept going and kept you going with it.
I'm actually thinking I should go and have it and try a hearing aid or something like that and see what it does really. And yeah,
it'd be interesting to have that sort of knowing so much about the science going on underneath. It must be interesting to be able to sort of put it to use kind of, or sort of every day.
Yeah, exactly. And hopefully maybe because of my interest in the knowledge that I've developed, I might be able hopefully to contribute something to the science of improving those hearing aids. They're quite tricky in a sense because they are working with an input system that is not working that well, and there's various strategies that companies are trying to improve the processing, and again it probably needs to be different for speech and for music, for example. So I'd be very curious to see if, for example, also music processing and enjoyment and appreciation can be improved in hearing aid users.
Great. Well, thank you very much for your time. It's been really interesting to have a chat about all these things. So yeah, just thanks very much.
My pleasure, and thank you for the great questions. And it was fun. Thank you.